Okay, sorry guys, I was just interrupted by a student. So back to this question. So this question is asking for our how we control our internal conditions, and one of the key ones that we need to keep the same is our temperature, and we need to keep our temperature within a set range for those enzymes. Okay, so to answer this question, you're just giving an example here, temperature. So I'm keeping temperature of my body constant or within a range of values, a very narrow range of values. Our body temperature can fluctuate between or plus or minus 5.5 degrees, okay, before something starts going wrong, before you start feeling poorly, okay? And that's why it's really important, if you have a temperature, to make sure you're getting that temperature down. And when people have children, it's really important, if they have a fever, a lot of the time, if it goes above a certain temperature, they have to go to hospital to get them to keep to try and get that temperature down, because babies, especially young babies, cannot regulate their body temperature when they're first born. Okay, so it's really important that you guys put other things in place as parents, okay, with those young children to regulate their body temperature, which is why babies are often covered in lots of blankets and things like that if it's cold outside. Okay, even if it's not that cold outside, they've got thick jumpers on and things like that to help regulate their body temperature. Okay, so the next question, multiple choice ones. A lot of you got these ones right. Okay, the organisation, levels of orders of organisation. Organelle is the smallest. Organ system is the largest. Larger than organ system, you have organism. Okay, this is a very, very small section of the specification, but you often get multiple choice questions or maybe tables asking you to put them in the correct order or giving you examples of each one. Okay. Yeast is an example of which group of organism? It is fungi. Okay, so yeast is an example of a fungi. That again is just memory recall and knowing your examples of your different kingdoms. Okay, if you can have one example for each kingdom, then that would be great. Okay. Now, question 4, okay, is the role of the organelles. Now, some of you made some silly mistakes here by mislabeling your organelles. So B is the nucleus, so I'm going to label them here. Okay. D is the cytoplasm. C is the vacuole. And A is a chloroplast. Okay. Now, you don't get any marks for correctly identifying what these parts are. Okay, the only bits you get the marks for are telling me what the function of the parts are. Okay, so even if you mislabel it, but you've got the function correct, and that is the correct function for that organelle that has been listed, you will still get the mark. Okay, which I think some of you, 2 of you, did that. Okay, maybe with the cytoplasm, you called it something else, but you gave me the correct function for the cytoplasm, therefore you got it. Okay, now you need to tell me the role of each of these. So chloroplasts absorb light to be used in during photosynthesis. If you mention chloroplasts are the site of photosynthesis, that will also be acceptable. Okay. The nucleus controls the activities of the cell or contains DNA. Any one of those will be correct for the nucleus. C is the vacuole, contains cell sap. Okay, you will also get the mark if you say it stores cell sap or stores sucrose or stores water or stores starch. Anything, okay, that is storage of in the plant will be acceptable there. And the cytoplasm is where chemical reactions occur. Okay, chemical reactions. That one's nice and easy. Okay, you only need to give me the functions, but some of you tripped yourselves up by getting the parts wrong in the first place. Okay, so always make sure. Some of you thought that A was a mitochondria instead of a chloroplast. Easy mistake to make, but if I've got a plant cell, which this is clearly is, chances are that's going to be a chloroplast rather than a mitochondria. Your mitochondria look like this. Look like little sausages with a squiggly line in it. They always have a squiggly line. Okay, I'm going to actually draw a mitochondria to show you what it looks like, because your mitochondria technically look like that. Okay, your mitochondria will always have those little squiggly lines in it because they have folds in their inner membrane, because that is where respiration happens. And if I've got more folds, I've got a larger surface area for more respiration to happen. Okay. So always assume that if it's a plant cell that it is going to be a chloroplast unless it looks like this. Okay, all that. Okay. Handy tip for you. Right, question 5. Now, this is where we start getting into the enzymes questions, and we start getting into the practical questions. These are the harder questions, okay? Now, we did say that we weren't going to answer this question. I did take that off, which is why your test was out of 48 instead out of 49 marks, okay? So, state what is meant by the term catalyst. Lots of you could tell me what was meant by the term catalyst. Some of you weren't specific though. Some of you said it speeds up an experiment or will speed up a practical. I need to specifically know that a catalyst will speed up a chemical reaction, okay? You don't even need to add on that it doesn't get used up by the reaction itself. That is more of a chemistry definition more than a biology definition. You can write that, you just won't get extra marks for it, okay? So it's only a 1 marker. We just want to know that it speeds up a chemical reaction, okay? And then you've got a bit of blurb here, and I'm going to highlight this for you, okay? Because these are the headings and the units that I need to have on my graph, because my next question is, okay, so I will come back to this in a bit, okay? But my next question is a graph question based on this table. But this practical is something that is similar to the one that we did. We did a question with potatoes, or a practical with potatoes, and this is a different take on it. So it's an enzyme question, and it says a teacher investigates the effect of enzyme concentration on the rate of reaction. So I automatically know if I'm investigating the effects of enzyme concentration, I'm changing it. So that is my independent variable. It's also in the left-hand column, okay, of a table. Anything in the left-hand column of a table is your independent variable, which means your left-hand column always goes on the bottom of a graph. Always, okay? All right. So my independent variable goes on the x-axis, the bottom, and my dependent variable goes on my y-axis. So a few of you did mix up your axes. Just be careful there because you will lose a mark for that, okay? He uses the enzyme catalase, which is found in potato. He changes the enzyme concentration by adding different numbers of potato discs. Catalase breaks down hydrogen peroxide into water and oxygen, and this is his method. He cuts the same size discs from a potato. He puts 5 centimetres cubed of hydrogen peroxide into each of 5 test tubes. Add a different number of potato discs to the hydrogen peroxide. Measure the volume of oxygen produced in 3 minutes. The teacher repeats this test 4 times for each concentration, and he calculates the mean rate of oxygen production for each concentration, and these are his results. So the question, the next one says, plot a line graph to show the effect of enzyme concentration on the mean rate of oxygen. This bit is important. Use a ruler to join the points with a straight line. Now, in biology especially, you will always have this. The only point where you won't have that sentence in the question is if it's asked you to draw a bar chart, okay? Now, this means that you are not going to get penalised by not doing your line of best fit differently from how you do it in maths. Here, it specifically asks you or instructs you in the question to join your points together with a straight line, okay? Now, you can also join it to 0. That doesn't make a difference. I haven't, just out of habit. If I haven't got a 0 value, I don't join it to 0, okay? So you will not get marked down for that in an exam as long as the rest of your graph is okay. Now, just a reminder that we mark for the scale. So the scale needs to be logical. Now, it doesn't matter if your scale on your y-axis is different to the scale on your x-axis, just purely because it was nice and easy. It was a nice and easy question because the units in my table were all they all went up to 10. So I just went up in 2s on both of them. If you went up in 2s on your x-axis and went up in 1s on your y-axis, that is fine as long as it's logical and it takes up enough space, Okay, they do say that your scale should take up at least 3/4 of your graph. Now, they haven't given you much graph paper, and I just used the lines that they'd already put at the side. Okay, but you could have drawn your lines further into your graph and had a slightly smaller graph as long as it wasn't tiny. Okay, you will get penalised for that. Now, you get a mark for your line. Okay, and that is basically us ensuring that you're able to read a question, because it says here, join your points together with a straight line here. You could also, I know it says with straight lines, you could also have drawn that as a curve if you were confident doing freehand line of best fits. Now, the bit of the curve that you would possibly do together is the bit here between 6 and 8, okay? The other bits you can draw with a ruler, or join together with a ruler. Now, your axes have to be the correct way round. And what I mean by that is you need to have enzyme concentration on the x-axis, as it is your independent variable, and you have to have your mean rate on the y-axis. Some of you got that the wrong way around. Your points, okay, are correctly plotted, and your units. Now, some of you were arguing with me why you didn't get 5 marks, and that is because you didn't put your units. Now, if I go back to the table here, my units, okay, are underneath here. So this is my units, they are in brackets, number of potato discs in brackets. That is what I need to have written on my graph, like I have here, mean rate of oxygen production in centimetres per minute, enzyme concentration, number of discs. If you didn't write both of those, I can only give you the mark for the units if you have written both of the units, okay, on your graph, on your axes, okay. All right, if you've only written one, all of you, okay, wrote down the mean rate of oxygen produced in centimetres cubed per minute, but not all of you wrote number of discs. I need both of them for you to get that mark. Okay, so that's why some of you have only got 4 rather than 5, but your graph looks exactly like what I've got here. Okay, but you're missing this bit here. Okay, so just be careful of that. Always put both units, okay, if it asks for it. Now, the next question asks you to explain, okay, the effect of increasing the enzyme concentration of the rate of oxygen production. Okay, now there's 3 marks here. Now, you have to explain this graph, okay? First of all, you have to describe it, okay? So I want you to tell me what this graph is showing me. So a good way to do is to start by describing it, and you get 2 marks for describing this graph, okay? It says, as the enzyme concentration increases, so does the production of oxygen, but it is not directly proportional. A lot of you stopped here and said that as the enzyme concentration increases, so does the production of oxygen, okay? Directionally proportional means that if I increase my enzyme concentration, my rate of oxygen produced is going to be increased by the same amount. And essentially, I will have a straight lined graph if that is directly proportional. And if I am making this statement alone, and only this statement, that is what I mean. That if I keep increasing my enzyme concentration, the volume of oxygen produced is going to increase as well. But that is not the case, because on my graph, I can see here that the rate of oxygen produced starts to level off. It starts to plateau. So I need to tell me that in my answer. So up to 8 discs where the enzyme concentration levels off. That is a description. That is 2 marks. You get 1 mark for telling me that as the concentration increases, so does the production of oxygen, but it levels off at 8 discs. Okay, after I've added 8 discs, the number or the volume of oxygen that is produced does not increase anymore. Okay, and then your 3rd mark comes for telling me why. Why do I see that? And that's what I've got here. That's this bit here. Okay, so it says, reason why is because there are more active sites available to react with the hydrogen peroxide until they are all full. So all of my active sites, when it starts to level off, will all be full. My enzymes can't take any more substrate, they can't break down any more substrate, so my rate of reaction cannot increase any further. It still happens, just at the maximum speed. Okay, think of it as if you're driving a car, some cars can only go up to 70 miles an hour, okay, regardless of how far you've got your foot down on the accelerator, okay, it can't go any further than it can, okay, so therefore it will keep going at 70 miles an hour until you stop it, okay, and that's what exactly is happening here. The rate of reaction isn't falling, it's continuing at that same speed because all of my active sites are full of hydrogen peroxide at that point in time, okay. And then we've got a bit of practical technique here. So we're looking at your practical and equipment knowledge. So I am relying on your chemistry knowledge a little bit here as well. Okay, so it says name a piece of apparatus suitable for measuring the volume of oxygen. You could have said a measuring cylinder, or you could have said a gas syringe. Okay, you need to correctly name the piece of apparatus. The only thing that I would have accepted is if you said syringe and not specifically gas syringe, I would have accepted that as well, because on the mark scheme, my gas is in brackets, which means that is an optional thing for you to write down. Okay. If you said anything else, I cannot accept that answer, okay, because it needs to be an sensible piece of equipment, okay, not a plunger. All right, so explain why it's important for the teacher to keep the volume and concentration of the hydrogen peroxide constant, okay. Now this is to do with the accuracy of your result. Now I can accept you saying that it needs to be a fair test, okay. That this needs to be a fair test for your results to be accurate. That is 1 mark. So to produce accurate results is 1 mark. And then the second mark is for telling me why my results need to be accurate. And the fact is that if I am changing the volume and the concentration of hydrogen peroxide, I am also going to be affecting the rate of reaction. Volume and concentration of substrate is a factor that can affect the rate of an enzyme control reaction. Same as if I change the temperature, okay? That is a factor that can affect the rate of reaction. So because of that, okay, I don't know whether my results will be accurate if I change the volume and concentration of hydrogen peroxide as well. So I need to control it, okay? So you get 1 mark for saying that we need to produce accurate results and 1 mark for saying because changing the volume of substrate would also affect the rate of reaction. Okay, that's where your second mark is here. Now, this one here, name another variable the teacher should keep constant in his investigation. The key word in this question is another. I've already talked about keeping the volume and concentration of hydrogen peroxide constant in the question before. Therefore, I cannot accept that as an answer here. Okay. I also cannot accept time because in the method they have also alluded to the fact that they are keeping the time the same here. So I cannot accept that either. I can accept temperature, I can accept pH. Okay, 2 other factors that I've talked about or that I know affect the rate of an enzyme control reaction. I can accept type of potato, I can accept size of potato discs, I can accept age of potato, okay, I can accept some other ones as well, okay, these are the main ones. I only want 1, okay, but most of you wrote these ones because you could see them and didn't think that because it said another, I needed to think of another variable that they haven't already mentioned, okay, and examiners are sneaky like this. This is where they catch you out, so be careful there, okay. Now this question 6, was quite well answered. The reason why some of you didn't get the full 4 marks is because obviously it was quite sneaky. I will talk you through the mark scheme, okay? You got a mark for saying specifically that diffusion is a passive process. If you described what passive means, so a lot of you said it requires no energy, but active transport does, okay? A lot of you said I gave you that mark, but you actually had to have passive, okay? You need to tell me it was a passive process and that it moves from high concentration to low concentration, whereas active transport doesn't. All of you could tell me that, so well done. I was really pleased with you, okay? Active transport does require energy, okay? But then most of you went on to then tell me that active transport also moves from a low concentration to a high concentration. If you've already said this marking point here, you've already said this marking point here, you can't get the marking point for telling me that active transport goes from low to high, okay, because I've already awarded that marking point here, okay. You need another difference between diffusion and active transport, and the one that I'm looking for here is this one here, that active transport requires carrier proteins for the process to work, but diffusion does not, and that's the mark that most of you were missing. Okay, so you were missing that marking point. Most of you got at least 2 or 3 here. You just missed some key bits, or the wording wasn't necessarily enough to get the marks. And that's all it is. Biology, you have to have these answers structured in a specific way to be able to get the marks. And that's where it's so frustrating. Okay, but that is what I am here to teach you. Okay, so do not get disheartened with this, because the more we do this, the better you will be and the higher your grades will go. Okay, so then we had this question, which was quite sneaky. It says substances move into and out of cells by a variety of processes, and it says explain the factors that affect the rate of movement of substances into and out of cells. Now here I'm expecting you to think about the factors that can affect membrane transport, which we did talk about, which was diffusion, osmosis, diffusion, osmosis, and active transport. All of those 3 things are membrane transport processes. Now, I want you to, in this question, explain all of the factors that can affect the rate of movement of substances. Now, we did this lesson as a table, and there were only 4 factors on there. There are 4 main factors, but there are some other factors in here that they have included, which is a bit sneaky because they've made it a 5 marker. Okay, so you have to not only tell me what the factor is, but you have to tell me how that affects the rate of movement. Okay, so some of you weren't getting the marks because you weren't linking it to the rate of movement for that substance. Okay, so the example I've got here is that temperature increases the kinetic energy of the molecules, which increases the number of collisions. This will increase the rate at which a substance is transported. That's 1 mark, that is. All of that for 1 mark. Each bullet point here is 1 possible mark. And I've got 6 bullet points here because I've given you an extra optional one that we didn't talk about in lesson today. Okay. The next factor that affects the rate of a membrane transport is the higher the concentration gradient, the higher the rate of movement. A shorter diffusion distance will also increase the rate at which a substance moves. A large surface area to volume ratio increases the rate of diffusion as there is less space to fill, and larger particles take longer to diffuse as they cannot pass through the cell membrane easily. That's the extra one I've added in. And increasing energy in the cell will increase the rate of active transport. Now here is the only one I have been specific on to a specific membrane process, and that is because energy is not required. Okay, it's not required for the process to work for osmosis or diffusion. They're passive processes, but active transport does require energy. So if I don't have a lot of energy in the cell, then active transport will not happen quickly. Okay, so that's why I have to be specific in that marking point. Okay. The next question was the 6 marker, and this was quite well answered for most of you. There were a few of you that misinterpreted what the question was actually asking you, and that's where you missed out on some of the marks. Okay. Now it's a describe question, so I don't need to tell me why anything happens. I just need to describe what does happen. Okay. And it asks you how named factors affect the rate of an enzyme catalyzed reaction. Now the named factors are temperature, pH, and substrate or enzyme concentration. Substrate or enzyme concentration. Okay. Now, because it's just said how named factors, I need to talk about all 3. And this is a question where I level it. So how I'm assessing this question, I am looking for a description of how each factor affects the name of, or affects the rate of an enzyme control reaction. You need to tell me how it affects the rate specifically. And to get 6 marks, I am looking for a detailed description of at least 2 of those named factors, okay? And a detailed description, I'm looking for at least 3 or more points for that factor and how that affects the rate of an enzyme catalyzed reaction, okay? So that is where some of you fell down because your answers were not detailed enough to get the marks, okay? And so therefore, to get 4 marks, you had to have at least 1 factor that had been described in detail, okay? Or 2 factors that had been described simply, okay? And because most of you have done all 3 simply, you got 4 marks, okay? So you need to add more detail to get those 6 marks, okay? So here, if the temperature of a reaction is too low, the particles will not have enough energy to move and collide. That's 1 marking point there. Enzymes must have, or most, have an optimum temperature of 37 degrees where they work the fastest. Okay, that's the 2nd marking point. If it is above the optimum, the enzyme can denature. That's the 3rd marking point, meaning the active site has changed shape, so the reaction will fall until it stops. That is the final marking point for temperature. Okay, that's all of the possible things that I could say about temperature. pH. Most enzymes have an optimum pH of between 6 to 8. That's 1 marking point. A pH either side of the optimum will cause the enzyme to denature. That's the 2nd marking point. And the rate of reaction will fall until it stops altogether. That's the 3rd marking point for pH. For substrate and enzyme concentration, if I increase the concentration, there will be a faster rate of reaction due to more collision. That's 1 marking point for concentration. Until there are no available active sites or the substrate runs out. That's the 2nd marking point for concentration. Now, to get 6 marks, I only need to have 2 of those detailed, okay, factors, okay? That is what I'm expecting you to write. That is a model answer for all 3 factors, okay, described in detail. Okay, to get 6, I only needed 2 of those. So some of you need to add a little bit more information to that question, okay? But well done, most of you answered that really well. And then this one, the lovely maths question, the last question, okay? Now, I did feel a bit mean about this because I didn't give you the equation, but it's never guaranteed that you're going to get it. So always prepare, okay? Because if you don't prepare and you assume they're going to give it to you and they don't, then that is an easy 3 marks that you're missing out on in your exam, okay? So what this question is asking you is to calculate the actual diameter of this cyanobacterium that they've given you a picture of, okay? And they've drawn across it the diameter of the bacterial cell, okay? They have said that it's magnified 50,000 times. Now, I want to calculate the actual size, so I'm going to have to rearrange my equation for magnification because magnification equals image divided by actual. So if I want my actual, I want image divided by magnification. Now, I have a line that I can measure to get my image size. So the first step that I've got is to measure the line A to B, and you get 1 mark for measuring that. You get 1 mark if you've written somewhere on your paper 6 centimetres, which all of you had. You all had 6 centimetres, so well done. Okay. It is easier if you measure it in millimetres going forward because you don't have to convert as much, okay? So if I'm converting 6 centimetres into micrometres, I need to multiply by 10,000 rather than just by multiplying by 1,000 if I'm converting millimetres into micrometres, okay? So it's just less work for you to do. So measuring in millimetres is going to be better for you if you're ever doing a magnification calculation. Now, I have to convert my millimetres into micrometres, okay, and I do that by multiplying by 1,000, so I've got 60,000. So I know my image size is 60,000 micrometres, okay? My actual size is my image size divided by my magnification, so I do 60,000 divided by 50,000 and I get 0.2. Now, what the common mistake that I saw here was either converting it incorrectly, so either converting it too much, okay, so I've got too many zeros, or converting it by not enough, so converting it by 100, not multiplying by 100, not 1,000, okay. The other mistake was rearranging the equation incorrectly, so a lot of you did image size times by magnification, okay, which is incorrect. That's if I'm working out my image size. I do actual size times by magnification, okay? So just be careful there. Show you're working out, because you can always pick up marks, okay? Please let me know if you want me to explain something in a little bit more detail or slightly differently if you still don't understand it, okay?